Hi all, welcome to the video for the module 5.1.1. This is going to be part of our introduction to bone tissue and what the quiz for this section is going to be on. Here, as always, you can see the learning outcomes and what will be expected of you. Please remember to use these for studying not only for your quiz, but for the exam at the end of the unit. All right, so the functions of the skeletal system can be seen here. There are 5 major ones, which include support, basically that the skeletal system provides a structural frame support for the entire body, and individual bones or groups provide a framework for attachment of soft tissues and other organs. Protection, which is just for delicate tissues and organs that are often surrounded by skeletal elements. The ribs protect the heart and the lungs, the skull encloses the brain, the vertebrae shield the spinal cord, and the pelvis cradles delicate urinary and reproductive systems. We have leverage. There are many bones of the skeleton that function as levers that can help to change the magnitude and direction of the forces that are generated by skeletal muscle. And the movements produce a range from the delicate motions of a fingertip to powerful changes in the position of the entire body. You have the storage of minerals. So the calcium salts of bone represents a valuable mineral reserve that helps to maintain normal concentrations of calcium and phosphate ions in body fluids. Calcium is the most abundant mineral in the body, and typically human body contains about 1 to 2 kilograms of calcium, with more than 98% of it deposited in the bones and the skeleton. And then finally, blood cell production, which is also known as hematopoiesis, and it's red blood cells, white blood cells, and platelets that are produced in the red bone marrow, which fills the internal cavity of many bones. All right, we're going to get into a couple identifications for skeletal classifications. So the adult skeletal system includes approximately 206 separate bones and a number of associated cartilages. We're not going to make you memorize 206 bones. That way of teaching A&P is antiquated. But we are going to cover some at least overall classifications. You will be able to recognize some bones when we get into the lab section, but as far as learning all 206 of them, I'm not going to make you do that in this lecture. This body system is divided into the axial and the appendicular skeletons, which we'll cover here. So the axial skeleton covers about 80 bones, consists of the bones that are in the skull, the hyoid, the sternum, the rib cage, the vertebral column, the sacrum, and the coccyx. And then your appendicular skeleton has about 126 bones, and this includes the bones of the limbs, and the pectoral and pelvic girdles that attach to the limbs to the axial skeleton. So bones are usually classified according to shape and structure, and also their surface feature. So flat bones can be classified as thin and roughly parallel surfaces, and flat bones form the roof of the skull, the sternum, the ribs, the scapula, and they also provide protection from the underlying soft tissues and offer an extensive surface known for, for the attachment of skeletal muscles. Sutural bones, also known as Wormian bones, are flat, small, irregular shaped bones between the flat bones of the skull. And these are individual variations in the number, size, shape, and position of sutural bones. Their borders are like the pieces of a jigsaw puzzle, and they range in size from a grain of sand to about a quarter. Long bones are relatively long and slender, and they're located in the arm, the forearm, the thigh, the lower leg, palms, the soles of the feet, the fingers, and the toes. The femur, the long bone of the thigh, is the largest and heaviest bone in the body. There are irregular bones, which have complex shapes with short, flat, notched, or ridged surfaces. The spinal vertebrae, the bones of the pelvis, and several of the skull bones, the mandible, for example, are irregular bones. You have the sesamoid bones, which are generally small, flat, and shaped somewhat kind of like a sesame seed. They develop inside of tendons and are most commonly located near joints of the knees, the hands, and the feet. Everyone has a sesamoid patella or kneecaps, but individuals vary in the location and abundance of other sesamoid bones. This variation, among others, accounts for disparities in the total number of bones in the skeleton. You also have short bones, which are small and boxy. And examples of these bones include bones of the wrist, known as the carpals, and bones of the ankles, known as the tarsals. So start to get into some of the gross anatomy of a long bone. Long bones are designed to transmit forces along the shaft and have a rich blood supply. So there are a few features that you'll need to know about these bones. And we'll start with the diaphysis, which is a long tubular shaft that forms the axis of a typical long bone. Then the walls of the shafts are made primarily of compact bone. You have the epiphysis, which is the ends of the bones that are composed primarily of spongy bones, also called trabecular bone. Spongy bone consists of an open network of struts and plates that are called tuberculae that resemble a lattice work with red bone marrow filling the spaces in between. The spongy bone is then covered by a thin layer of compact bone and articular cartilage. And there are two things that you'll need to know about the epiphysis. You have the proximal epiphysis and the distal epiphysis, which are just the proximal is the end closest to the origin of attachment, and the distal is the end furthest from the origin of attachment. You have the metapiphysis, which is a narrow zone that connects the diaphysis to the epiphysis, and the episioal plate is a thin layer of hyaline cartilage that is more commonly known as the growth plate and is important for the growth of the long bones. You also have the medullary cavity. So within the shaft of the long bone, there's a cavity where the bone marrow is located. In childhood, the medullary cavity is filled with red bone marrow, but as we age, fat accumulates inside this red bone marrow and helps to transform it into yellow bone marrow. Red bone marrow is important for hematopoiesis, but yellow bone marrow is no longer hematopoietic and instead stores fat as an important energy source. There are a couple membranes that are associated with bones as well. You have the periosteum, which is the outermost covering of bone, which is made primarily of dense irregular tissue and held on by some collagen fibers. You have the endosteum, which is the internal membrane of bone made of connective tissue. It also lines many of the canals that pass through the bone to help support and supply blood and nerves to the bone. You have the nutrient foramen, which is in order for bones to grow and be maintained, they require an extensive blood supply. And the nutrient foramen is a tunnel that penetrates the diaphysis to provide access to the blood vessels to the shaft of the bone. Within the nutrient foramen, you have the nutrient artery, which transports oxygenated rich blood to the bone. And then you have the nutrient vein, which transports deoxygenated blood and waste-laden blood from the bone. You have the metaphyseal artery and the metaphyseal vein, which carry blood to and from the area of the metaphysis and the epiphysis. You have articular cartilage as well, which covers the portions of the epiphysis to articulate with other bones. The cartilage is avascular, so like hyaline cartilage, and provides primarily a diffusion from the synovial fluid to maintain oxygen and nutrients to eliminate wastes. There are several bone markings that we'll get through here. We will cover a little bit of this in the lecture portion, but most of these you'll be responsible for as we transition to this in the lab. And you can see all the examples here. You don't need me to read that to you. As far as compact bone is concerned, there are a few features that are related to this. So some of the functional units of a compact bone is the osteon, which is the basic structural and functional unit of bone, consisting of bone cells that are organized around a central canal and separated by concentric lamellae. You have the central canal, which is also known as the Hebnerzian canal. It runs parallel to the axis of bone and is typically located in the middle of each osteon. Each central canal possesses an artery and a vein, a lymph vessel, and a nerve. You have the perforating canal, which is a passageway that extends perpendicular to the axis of the bone and connects the central canals to the adjacent osteons. You have the lamellae, which are nested in concentric rings of matrix around the central canal. And there are two types of lamellae that you'll be responsible for knowing. You have the circumferential lamellae, which are specialized lamellae found at the center and inner surfaces of bone, where they are covered by the periosteum and endosteum, respectively. So these lamellae are produced during the growth and maintenance of the bone. And then you have the interstitial lamellae, which fill the spaces between the adjacent osteons of compact bone. These lamellae are remnants of osteons whose matrix components have been almost completely recycled by the actions of bone-digesting cells. You also have lacunae, which are mature bone cells, often called osteocytes, that are trapped within an open space called a lacuna. Osteocytes cannot divide, and therefore each lacuna consists of only one type of osteocyte. You also have the caniculi, which are processes of the osteocytes that extend into narrow crevices called caniculi. that penetrate the lamellae and connect the lacunae to the central canal. Bone is also associated with 4 types of cells that account for approximately 2% of that bone's weight as well. You have the osteocytes, which are mature bone cells that maintain the protein and mineral content of surrounding matrix through the turnover of the matrix components. Osteocytes secrete chemicals that help to dissolve adjacent matrix and release minerals into their circulation. The osteocytes then help to rebuild that matrix, stimulating the deposition of mineral crystals. Osteocytes also participate in the repair of damaged bone. You have osteoblasts, which are immature bone cells that are located on the surface of bone that help to produce new bone matrix in a process called osteogenesis or ossification. Osteoblasts make and release the proteins and other organic components of the matrix. Before the calcium salts are deposited, this organic matrix is called an osteoid, and osteocytes develop from osteoblasts that have become completely surrounded by bone matrix and trap them within a lacuna. In addition to those two, you have osteoprogenitor cells, which are mesenchymal cells located within the periosteum and the endosteum. These stem cells help to divide to produce daughter cells that help to differentiate into osteoblasts and are important for the formation of osteocytes. And finally, you have osteoclasts, which are considered bone digesting cells that remove and recycle bone matrix. These are giant cells with more than 50 or more nuclei. Osteocytes are not related to osteoprogenitor cells or their descendants. Instead, they're derived from the same stem cells that produced phagotic white blood cells called monocytes. Acids and proteolytic enzymes secreted by osteoclasts help to dissolve the matrix and release stored minerals that are within bone. And this process is called osteolysis or reabsorption, and it's important for bone remodeling. All right, we're going to get into the formation of bone or ossification. Basically, what happens is bone replaces hyaline cartilage, and we'll continue through that here. So we have 2 different types of bone formation that we'll talk about: intrachondral ossification and intramematous ossification. So in intrachondral ossification is going to be seen as the formation of that hyaline cartilage molecule, and it begins approximately at 6 weeks after fertilization. The hyaline cartilage does not turn into bone, but instead is broken down as ossification occurs. And there are a few steps of intrachondral ossification. You have the cavitation of hyaline cartilage, which you can see in the first couple pictures of that cycle. Basically, what happens there is the chondrocytes within the shell or in the shaft of those bones hypertrophy, which means they enlarge, and the surrounding matrix begins to calcify. And the impermeable matrix causes chondrocytes to die from lack of nutrients, leaving the matrix that starts to deteriorate. Blood vessels then grow around the edges of the cartilage, and the cells of the perichondrium convert the osteoblast, producing a superficial layer of bone, sometimes called a bony cartilage. Step 2, you have the invasion of the internal cavities. In this section, blood vessels penetrate the cartilage and invade the central region. This area within the shaft of the hyaline cartilage is called primary ossification center. And then migrating blood vessels are called fibroblasts, which differentiate into osteoblasts eventually. Lymph vessels, nerves, red bone marrow fragments, and collectively these are all called the periosteal blood. The osteoblasts secrete osteoid around the remaining fragments of hyaline cartilage that form the trabeculae or spongy bone. In the third stage, you have the formation of the medullary cavity. And that starts to form here. As the primary ossification center enlarges, osteoclasts break down newly formed spongy bone and open up a medullary cavity in the center of the diaphysis. The osseous tissue of the outer shaft then becomes thicker and forms compact bone. In the last stage, you have the formation of the epiphysis. So secondary ossification centers start to appear in the area of the opposite ends of the bone. The cartilage in the epiphysis calcifies and then deteriorates. That forms the cavities that allow entry of a periosteal blood. Soon the epiphysis are filled with spongy bone, and the spongy bone is not broken down during the remodeling process. Contrary to that, you can have intramembranous ossification. This is the formation of bone without the cartilage monocle. Typical in flat bones, mandible, clavicles, and the patella. This begins approximately 8 weeks after fertilization. You have mesenchyme cells that differentiate into osteoblasts and within fibrous connective tissues. And this type of ossification normally occurs in the deepest layer of the dermis or in the connective tissues of the tendons. And there are a few steps of intramembranous ossification. You have the formation of the bone matrix within the fibrous membrane, in which mesenchymal cells cluster and secrete organic components of the matrix. And the location of this activity is the ossification center. This results in the osteoid mineralizing and the mesenchymal cells differentiating into osteoblasts. And as ossification proceeds, the osteoblasts get trapped within the lacunae and differentiate into osteocytes. Second, you have the formation of woven bone in the periosteum. The osteoid accumulates, fuses together, and formulates struts called trabeculae, or spicules, around the blood vessels. The overall structure is similar to spongy bone in this situation. The formation of compact bone plate is the third and final step. And initially, the intramembranous bone consists of spongy bone only, but subsequent remodeling around the trapped blood vessel can produce osteomes typical of compact bone. And as the rate of growth of the bone slows at the surface, the connective tissues around the bone become organized into the fibrous layer of the periosteum. In addition to this, we can have bones growing. This occurs by two methods: longitudinal growth, which is responsible for length, and in longitudinal growth, hyaline cartilage cells form tall columns at the epiphyseal plate, or the growth plate, and within the articular cartilage. The cells at the top of the stack then divide quickly and increase the thickness of the epiphyseal plates, causing the entire bone to lengthen. Older chondrocytes closer to the shaft enlarge, die, and the surrounding cartilage matrix then deteriorates. The deterioration leaves spicules of calcified cartilage, and then osteoblasts in the medullary cavity then ossify the cartilage spicules and help to form spongy bone. The hyaline cartilage at the epiphyseal plate is eventually replaced entirely by bone, and then once completely replaced with bone, the epiphyseal plate is now called the epiphyseal line. And this typically occurs in person's early 20s, and as a result, the person stops growing in height. In addition to longitudinal growth, you have appositional growth, which can be considered widening of the bone. And in appositional growth, the osteoprogenitor cells beneath the periosteum differentiate into osteoblasts and form new osteons in the external bone surface. While the bone is being added to the outer surface through appositional growth, osteoclasts are removing and recycling lamellae in the inner surface. And as a result, the medullary cavity gradually enlarges as the bone increases in diameter. Appositional growth is important in increasing the diameter of existing bones, but does not form the original bone.